our own, here at bioengineering. And she's going to be talking about engineering 3D lung models to enhance early stage drug discovery and validation. Yeah, so with that, Chelsea, I guess she said she's okay with having questions throughout the seminar. So if you guys have questions, you can save your question or write it down on the chat, and then Tammy will read it for Chelsea. Or if not, you can just wait until the end. It's not a problem. You can do either way. All right? So without further ado, please, Chelsea, you might go ahead and share your screen and start your seminar. Thank you so much.
Well, thank you so much for the introduction. I'm excited to be here with y'all on Pi Day. I hope you all get to celebrate. So I'm thrilled to share our work on engineering 3D models of lung tissue to enhance early stage drug discovery and validation. I like to start by getting everyone thinking about lungs. So please take a deep breath with me. Inhale, exhale. You probably don't realize it, but you breathe 22,000 times every day. That's over 8 million times every year. It's a lot. And for someone with a chronic pulmonary disease, they notice every single breath that they take. These diseases cause shortness of breath, chest pain, heart palpitations, dizziness, fatigue. Most of them are progressive and irreversible. And our current treatment options can relieve the symptoms, but they don't cure the disease. Transplantation is our only option. I used to have a generic picture of someone from the internet right here. But at the end of 2022, I was diagnosed with asthma. I lived for nearly 40 years not knowing I had a chronic pulmonary disease. And this picture is from a hike I did in Patagonia where I had to stop the group over and over again to catch my breath. Now that I have a treatment, my symptoms are better, but the underlying condition will be there with me for my entire life. So it really motivates me as an engineer to try to create new models for us to study these diseases. Right now, traditional models that we have, like growing cells on a flat Petri dish or doing tests inside a mouse model, have taught us a lot about safety and efficacy of treatments, but they don't fully recapitulate what's happening in a human. And for that reason, over 90% of the drugs that are successful in animal models fail in human clinical trials. So my vision is that using dynamic 3D multicellular systems, we can accelerate the early drug discovery and validation and hopefully pick out drugs that work better in a human once we get there. These fully human cellular systems will improve our ability to predict, replace, and refine animal testing. Custom biomaterials allow us to use iterative improvements to test our designs and keep testing until we have the right design. And something that just came out that's really exciting is that the FDA no longer requires animal models for every single drug application that's submitted. So US FDA scientists are now working to establish quality controls for accepting data from 3D multicellular systems instead of animal models. Before I dive more into my work, I wanted to tell you a little bit about my journey to the point that I'm at now and give you a little insight into how I'm thinking about potential commercialization of these products. So I started out in material science and engineering. I continued on to get my PhD in biomedical engineering. I stayed at the University of Florida for both degrees, and part of that was because I was a first-generation college student. University of Florida was close to home, and I didn't know a lot about going to graduate school, and neither did my parents. I completed a postdoc at CU Boulder in 2013, and then I became the director of product development at Sharklet Technologies. I worked there for 4 years before joining the faculty here at CU Denver Anschutz. So during that time, I did an internship between my junior and senior year of my undergraduate. And the first day I started at Kimberly-Clark Corporation, I was handed an invention disclosure. What is an invention disclosure? It's a piece of paper that you fill out when you are working at a company or a university and you think you have an awesome idea that you'd like to patent. So someone else had filed this invention disclosure and then they handed it to me, the summer intern, right here on the softball team, and told me, please reduce this to practice so we can file a patent. Well, what does that mean? I spent my entire summer internship making a prototype of what we eventually filed a patent on, which is this wipe device for applying baby lotion or diaper rash cream without having to touch it. So it's sort of a single-use wipe that you would open up this plastic inside, there would be lotion on this wiper part, and you could apply it without getting diaper rash cream all over your fingers if you were out traveling. Over my 2 internships at Kimberly-Clark, we filed 3 patent applications, and this was my first experience with intellectual property, filing patents, and protecting our ideas so that we could commercialize them later. And then I mentioned that I did my PhD at the University of Florida. My advisor, Tony Brennan, worked in industry before he became faculty, similar to the way I did. And so we actually talked about intellectual property in an academic setting throughout my training. And we always made sure to file patents before we publish papers, which I don't think is common in academia because we know papers are our currency. So I wanted to mention it to the students watching today so that you can think about this process as you're creating and inventing new things, especially in engineering. It happens a lot. Then I worked at a startup company and had even more opportunities for invention. So at the startup company, you can see I filed over 13 US patents and something like 75 additional patents across the globe. We spent a lot of time innovating and just reminded me to always protect our inventions because some of these things later went on to be commercialized and started the process of being turned into a product. Now, as an assistant professor, we talk about this in my lab a lot, and we have filed 3 provisional patent applications that are in process working with CU Innovations. And so when you're able to protect your intellectual property from your laboratory, you'll have the opportunity to potentially commercialize it later, which is what I'll talk to you a little bit about in addition to our research. So back to the research, what are we studying? Pulmonary fibrosis is the main lung disease that I'll talk to you about today. It's progressive and incurable, and it's what we call a restrictive lung disease. So the volume of your lungs that is able to breathe in air is actually reduced. And this happens because there's scarring across the gas exchange surfaces or alveoli in your lungs. You can see from these chest x-rays over time where fibrosis is marked in red that this disease is progressive and starts to spread throughout the body or throughout the lungs. On this side, you can see 2 histology images, and one of healthy lung, where you see a very thin single cell layer of epithelial cells around these gas exchange surfaces or the alveoli in your lungs. And then in a fibrotic lung, you can see that the cells have proliferated. There are many more cells there than there should be. Some of these are epithelial cells, and some of these are connective tissue cells called fibroblasts. So as this scarring continues, it's very much like scar tissue. It begins to stiffen your lungs and make it harder and harder for you to breathe. So the vision in my laboratory is that we can use tissue-informed biomaterials to study lung disease and repair. So this image is, can see a little bit similar to the histology I showed you, where we have wide open lung spaces surrounded by cells. And this was created using biomaterials and a 3-dimensional model. And I believe in this model, we'll be able to study cell-cell interactions, cell matrix interactions, and sex differences, which are all important components of chronic pulmonary diseases. So let's talk a little bit about materials first. There are many different types of biomaterials. The ones that are most commonly used to study the lung are naturally derived biomaterials. So if anyone's in the materials field on the call today, you've probably all heard of Matrigel. Matrigel is a material that they actually pull out of tumors that are grown in mice. So they take all the cells out, and then all of the proteins and growth factors and other materials that are left over become a substrate that scientists grow cells inside of. This is great. It works really well. Cells like to interact with all of those signals that they get from the tissue that used to be a tumor. However, it's very hard to control. We can't control the mechanical properties of the material, and we can't change the material to make it better because we don't even know exactly what's in it. On the other end of the spectrum is a fully synthetic biomaterial. So these are materials like polyethylene glycol or PEG that start out as a blank slate. We can add in specific cues that would interact with cells, like a short peptide sequence that would bind with integrins, or we could add in specific cross-linkers that are degradable by enzymes the cells secrete. So this reductionist approach allows you to understand and to study 1 or 2 interactions between the cells and the extracellular environment at a time. That's fantastic for working in reductionist systems, and it allows you to control the mechanical properties. However, there aren't as many endogenous cues for the cells, and we can't learn as much about the biochemical composition of the surrounding material. So my lab has pioneered using hybrid hydrogels. These hybrid biomaterials contain both proteins and growth factors from the lung tissue and a synthetic backbone that allows us to dynamically control the mechanical properties. So now we kind of get the best of both worlds. We can understand the mechanics and we can study the biochemistry as well. So the first thing we do to make three-dimensional models of lung is to create a template for making that 3D structure that I showed you. We create cell-degradable microspheres to recreate 3D distal lung structure. These microspheres are made out of polyethylene glycol. This is a hydrophilic material that absorbs quite a bit of water, and we're able to tune the stiffness of this material to match your lungs. We include a crosslinker that's degradable by enzymes that the cells make. We add in a fluorophore so that we can see it under the microscope. And then we add in short peptides, including RGD and YIGSR. These mimic the proteins in the lungs, fibronectin and laminin, and it helps the cells stick to the microspheres. We've also included magnetic nanoparticles because part of our process to create the 3D lung structure is to aggregate these together into a cluster to mimic the alveolar tissues. So to make the microspheres, we do an emulsion polymerization. This is just like salad dressing. When you shake up that bottle that has oil and water in it, and you get those little spheres that you can see in the salad dressing, that's what we're doing here. We spin the material quickly. We have an oil phase, and then inside the aqueous or the water phase is all of our material components. This creates small microspheres that we can then polymerize using light. And then at the end, you'll see they look like this. We filter the microspheres to make sure our average microsphere is about the same size as a human alveolus, which is 200 microns in diameter. And then we did some testing to make sure the elastic modulus or the stiffness of our hydrogels was within the range of healthy lung tissue, 1 to 5 kilopascals. These microspheres can be degraded in the presence of cell-secreted enzymes, and I just wanted to show you a demonstration of that here. So we added in enzymes to our cell culture mixture, and you can see that we start out with microspheres at 0 minutes, and these degrade over time, and it's shown here in the video as well. Our hydrogel microspheres are first coated with epithelial cells. These are the cells that line the airways and breathing spaces in your lungs. And then we use the magnets that we put into the microspheres to aggregate this structure together to create something that looks like lung tissue. Then we encapsulate this whole cluster of cells and microspheres into an embedding hydrogel. And this embedding hydrogel can be many different types of hydrogels. The data I'll show you today, we embedded it in a stiff hydrogel that mimics fibrotic lung tissue, or a soft hydrogel that mimics healthy lung tissue. We could also embed these in our hybrid hydrogels. We'll chat a little bit more about that later. So the encapsulating hydrogel was again based on polyethylene glycol. This time we have a cross-linker that's degradable by the fibroblast. And again, we have these two peptides that help the cells stick to the materials. These are just, oh, Kim.
One question came up. What instrument do you use to do the tensile test?
Oh, that's a great question. We are doing our mechanical properties. We're not using a tensile test because it would break our hydrogels, but we're doing our mechanical properties testing using a rheometer. So it applies a rotational shear stress to the material, and then we can measure how the material responds to that force. Great. So after we have all that situated and we apply our magnets to the culture, we watch them aggregate. So you can see here, the microspheres look kind of like a white opaque color, and the cells have magnets on them. So you can see them here in this kind of rust colored dots. And over time, over 3 days of aggregation, the cells begin and the microspheres begin to cluster together. And then if we take microscope images of that, it looks a lot like lung tissue, which was really exciting. So then we encapsulate everything inside a supporting hydrogel, and you can see this is just a confocal 3-dimensional image at week 0 that the cells labeled in green are sticking to the microspheres that are red, and that it's happening in 3 dimensions. Next, we wanted to understand more about what the cellular responses to being in this microenvironment were. So we show here that 3D lung models supported cellular viability for up to 3 weeks. We have both the epithelial cells in the middle of the construct and the fibroblasts around the edges. So we used a live dead stain where live is green and dead is red to identify the percent live cells in the constructs. And you can see about 80% of the cells are alive throughout the 3-week long culture, which is a pretty long time to culture cells. And then the representative images down here are showing the live cells in green, like I mentioned, and the red cells are dead. And again, just a really nice demonstration of this architecture we're able to build using the microscopes. So in order to study fibrosis, we needed to include both of these cell types, the epithelial cells that line the breathing spaces and the fibroblasts, which are the support cells around the breathing spaces that tend to migrate into and take over and form scar tissue during fibrosis. So in order to study this process, we used a dual transgenic reporter mouse. And the cells from these mice allow us to monitor fibrosis over time. So this is a CT scan of a healthy lung. This one's fibrotic. You can see there's much more dense areas of collagen deposition in this fibrotic lung. And then these are the reporter cells. The reporter cells will light up in red when alpha smooth muscle actin is produced and green when collagen 1 is produced. And both of these proteins, when they're expressed, are markers of fibrotic activation. So the data down here are just showing that we're able to digest these lungs and sort out the cells. We get, when we're aiming for an epithelial cell population, we get 100% epithelial cells, and most of those are alveolar type 2 epithelial cells, the epithelial cells that are in the air spaces. And then when we are sorting out for fibroblasts, we're getting 100% fibroblasts that are making vimentin and this PDGFR alpha receptor, which means that those fibroblasts are located next to those epithelial cells in the lung. So next, we put these into our constructs around our AT2 cells, and some of our tests were on fibroblasts only, and some of our tests included the AT2 epithelial cells. And then we use these dual transgenic reporter fibroblasts to look at how the cells are activating in 3 dimensions. You can see at week 1, we don't really have very much fluorescence apart from the blue stain here that's marking the nuclei of the cells. Week 2, we start to see more activation, especially in the stiff microenvironments. And then in week 3, we see the most activation again in the stiff microenvironments. When we quantified this activation, we were expecting to see more activation in the stiff microenvironments, but we were surprised to see the very most activation that we measured was in an environment that also included epithelial cells. So we started to think more about this and wanted to ask the question, well, what are these epithelial cells doing to make these fibroblasts respond in that way? So these counts are the number of cells that are activated that are either positive for collagen 1, alpha smooth muscle actin, or both. And then we looked at the percentages of each of these different configurations. So you can see in the early weeks that we have higher amounts of collagen 1 expression, and this tends to go down, and we have increased amounts of alpha smooth muscle actin expression after that. But how do the epithelial cells interact in this microenvironment, and what role are they playing? So the alveolar type 2 epithelial cells are a progenitor cell in the lung. And when the lung is injured, they can differentiate into alveolar type 1 cells. Those are the very long, thin cells that actually do the gas exchange. These alveolar type 2 cells create surfactant protein C, which is labeled in this peach color. And then transitional cells in the mouse model start to create keratin 8 proteins. When these cells actually differentiate into AT1 cells, they create potoplanin in blue. So we can see over time, especially in our stiff microenvironments, our AT2 cells are transitioning into AT1 cells. And when that happens, these cells produce a molecule called TGF-beta, which can activate the fibroblasts. So having these models where the AT2 cells are transitioning into AT1 cells like they would in an injured fibrotic lung, activating the fibroblasts, we found that it was necessary to have both cell types in order to actually mimic the disease. We took a deeper dive and looked at gene expression. So we saw that cell-cell and cell matrix interactions influence gene expression. We saw changes in inflammation, extracellular matrix remodeling, integrin signaling, proliferation, and TGF-beta signaling. And so all of these gene expression changes just confirmed that we were seeing differences that were trending toward fibrotic gene expression in our fibroblasts and epithelial cells in the stiffer microenvironments. So where do we take this from here? One of the things we've been working on in our laboratory that I mentioned before was hybrid hydrogels. These hydrogels can include proteins from healthy or fibrotic lung tissue. So for this example, we've used healthy or bleomycin-injured mouse lung tissue. The bleomycin injury creates a fibrotic response in the mouse lungs after 21 days. We take these tissues and then we expose them to a sequence of detergents and DNase to remove all of the cells and cell material. And then we homogenize and pepsin digest to create decellularized extracellular matrix. So these are all of the proteins, growth factors, and other support molecules that are in the lungs that are surrounding the cells. You can see when we treat these mice with bleomycin, if you look at readouts from lung function testing, you can see decreases in the total lung volume that relate to and show that we're doing injury to the lungs and that the fibrosis in the lungs is preventing the mice from breathing as they would normally. In this figure, we show intact lung tissue has many, many cells in it by staining with DAPI for the nuclei. And then when we decellularize the tissue, we see virtually no cells. We also confirm decellularization by looking at the concentration of double-stranded DNA that was left in the tissue. You can see that both healthy and bleomycin after decellularization are below the cutoff of 50 nanograms per milligram. Next, we have incorporated these molecules, these decellularized extracellular matrix proteins, into a dynamic hybrid hydrogel. So these hydrogels can be tuned to recapitulate healthy or fibrotic lung mechanics. And the really cool thing about these materials is that we can now decouple these two things that are intrinsically linked inside an organism. When a lung becomes fibrotic, it starts to create more and more collagen. As the lung creates more and more collagen, it's changing the biochemical composition of the extracellular matrix. And it's also changing the mechanical properties. So as more and more collagen is expressed, the lungs become stiffer and stiffer. And these two things can't be decoupled in vivo. In our material system, we can decouple these things. So first, we use a chemical reaction to treat the decellularized extracellular matrix molecules, and we convert the primary amines on these proteins into thiol molecules. These thiols can react with our polyethylene glycol alpha methacrylate molecule. If anyone is a chemistry nerd, you'll note that this alpha methacrylate is connected to the PEG backbone on the opposite side of the alpha carbon. This allows the molecule to degrade by releasing this small alcohol group and not breaking the backbone of the polymer. So our polymer is stable over at least 60 days in cell culture, which allows us to run long-term models of chronic disease. When we combine these two materials using a Michael addition reaction, so we just increase the pH a tiny bit to be basic, we can create a soft hydrogel. This hydrogel can mimic healthy lung tissue. And then we can stiffen the hydrogel by exposing the whole system to a photoinitiator and 365 nanometer lab. This initial reaction is done off stoichiometry. So that means that we leave some of these alpha methacrylate groups open, and then later we can react them to stiffen the hydrogel. So in that way, we can create healthy soft hydrogels, healthy stiff hydrogels, which don't exist in vivo, diseased or fibrotic soft hydrogels, which again don't exist inside a mouse model, and stiffened fibrotic hydrogels. So now we can decouple the biochemical changes in the lung from the mechanical changes in the lung and start to understand which one of these is a bigger driver of fibrotic disease progression. Again, we used our primary mouse fibroblasts that have this dual reporter system, and we saw that levels of activation in response to stiffening were higher than they were in response to just biochemical cues alone. So the data in the graph down here shows the percent of activated fibroblasts when grown in these microenvironments. You can see on the soft hydrogels, whether they're healthy or or filled with decellularized extracellular matrix from a fibrotic mouse, their levels of activation are relatively low, around 40%. Upon stiffening, the levels of activation for both of these microenvironments increases to about 80% with no significant differences. If we normalize the fluorescence intensity per cell, so if we say, okay, we're not just looking at whether the cell is making this or not, we're actually looking at how much of the protein the cell is making, we see a very similar difference, but we start to see a trend toward increasing activation in response to the biochemical cues. Overall, we believe that in this model, these results are showing us that the mechanical cues are having a bigger influence on cellular activation than the biochemical cues. The really cool part about this system is that our secondary polymerization reaction is controlled by light. So that means we have control over this reaction in space and in time. So I just wanted to show an example of that. These materials here, we incorporated a fluorophore into the secondary reaction so you can visualize it. These are 50 micron by 50 micron stripes. And you can see this purple stripe here is a stiff area, and this dark stripe here is a soft area. So when we look at cellular activation in response to these patterns that we can draw, you can see that 50 microns wasn't quite wide enough to encapsulate an entire cell. So these cells were feeling both soft and stiff microenvironments. But when we got to 100 microns, we could see this cell is all by itself inside a stiff stripe, and it is lit up with both green and red, showing it's very activated. So our results with the proportion of activated cells, you can see there aren't big differences between soft and stiff in the alpha smooth muscle actin readouts, but we're starting to see them in the collagen 1 readouts. With this proof of principle in 2 dimensions, we're starting to move this work into 3D, because when a person has fibrosis in their lungs, it creates small areas that have dense activated fibroblasts and dense collagen that are called fibrotic foci. We're really interested in understanding the size and shape of those foci in human lungs, and then recreating them in 3D to understand how the cells inside, at the interface, and outside of those areas are responding to these changes in stiffness in the microenvironment. It could be that the cells that are nearby these activated areas become activated, and that's one way that the disease spreads throughout the lungs. Just a couple slides here to show you that we've done a lot of work in mouse, but I told you at the beginning that mouse work does not create the best candidates for drugs in humans. So we were also working on creating fully human systems. We have been able to successfully treat human DECM in the same way that we treated the mouse decellularized extracellular matrix from lungs. And we have incorporated that into our hybrid hydrogels as well. So you can see there's a picture down here of synthetic hydrogel that just looks light pink when you stain it with picocereus red, a stain that highlights collagen fibers. But in the hybrid hydrogel, you can see this much darker red staining that indicates that we have incorporated collagen into the hydrogels. And collagen would be the main component we expect from decellularized extracellular matrix. Again, we grew these cells on our soft hydrogels and growth media for a couple days. Then we switched over to our activation media, which is just a lower concentration of serum. And then on day 6, half our samples were collected as soft samples, and the other half were stiffened, and then those were collected for analysis on day 9. So you can see in the stiffened human hybrid hydrogels, human fibroblast activation increased on the stiffened microenvironments. You can see here in staining for F-actin and alpha smooth muscle actin, and also quantified below. The cells increased in aspect ratio when we stiffened the microenvironments, and also increased in activation or expression of that alpha smooth muscle actin. So we're excited. This paper was just accepted at ACS Advanced Materials and Interfaces, and should be out within the next week or so. The next step we're taking to make these fully human models is using induced pluripotent stem cell derived cells. So iPSCs, or induced pluripotent stem cells, are cells that we've been able to take from a human adult donor. So we could take a skin biopsy, we can reprogram the cells to become stem cells, and then we can differentiate those stem cells to become lung cells. When we have those lung cells, we can incorporate them into our hybrid hydrogels, and you can see Here is a spheroid of those lung cells. They're starting to grow and mimic the cells that line the breathing surfaces of your lungs. They're surviving inside our hydrogel materials. This is the first time that we know of that anyone has grown these cells in 3D in a material that wasn't Matrigel. So we can show that tuning our biomaterial and changing the crosslinkers allows us to create spheres that have the same size as human alveoli and have good sphere quality and total sphere number. If we incorporate what we've done before, so we put these iPSC-derived epithelial cells into a hydrogel that is either soft mimicking healthy lung or stiff mimicking fibrotic lung, along with fibroblasts, we can induce gene expression changes that start to mimic fibrosis, especially in the stiff microenvironments when we add TGF-beta, which is an inflammatory molecule that activates the fibroblasts. So we can see here, if we're looking at the fold changes of these genes, this is collagen 1, and ACTA2 is the gene that controls alpha smooth muscle actin. We see significant increases when we add TGF-beta in the stiff microenvironments. We also see changes in fibronectin production, latent TGF binding protein 2, and then also CTGF, which is a measure of a growth factor, connective tissue growth factor. So all these changes indicate that both our microenvironment and the biochemical cues that we're giving the cells allow us to create a fibrotic model using human cells and human tissues. Something else that I'm really excited that we're working on that I wanted to point out, because pulmonary fibrosis is twice as likely to happen in men than women, and we don't quite understand why yet. Some epidemiologists will tell you, oh, it's because guys are exposed to different environmental hazards. But on the biological level, we're not sure that's true. And we're really excited to start to study that in more detail. So this is some work from Michaela Mueller in my lab, and she has been studying the differences between female and male fibroblasts and what type of serum we add to the media. So traditional growth media for all of our cells contains fetal bovine serum. So this is serum from the blood of young cows. And these young cows' blood is harvested usually either while in utero or right after. So they have high levels of progesterone and other hormones that are related to females usually. So we tested our activation of these cells in a sex-specific way, and we either matched female cells with female human serum or male cells with male human serum, or we mismatched it. We did the same experiments where we started all of our cells on soft microenvironments, and then we stiffened some of the microenvironments to look at how the cells were responding to increases in mechanical stiffness in the microenvironments. So you can see from these results that were just performed in the fetal bovine serum, the FBS, that female cells didn't see a big increase when the microenvironment was stiffened. They seemed less responsive, but overall seemed to be slightly more activated. The male cells, we see this difference between soft and stiffened that we might have expected. When we ran all of the experiments using female and male serum, and we also tried out different ages below and above 50, and looked at how these cells are responding to the different sex hormones and different concentrations of the sex hormones in the media, we saw some really fascinating trends. First, we saw what we expected, that activation of our cells overall increased on stiffened microenvironments. Then we saw that activation of these cells also increased as the age of the serum increased. And this also goes along with pulmonary fibrosis. It tends to be diagnosed in older patients. When we looked at the different responses to increasing hormone concentrations in the human serum that we tested, we saw that percent activation increased with increasing amounts of hormones in our different serums. And we saw a trend that's important for pulmonary fibrosis, that these fibrotic activation increased with increasing testosterone. But you only see this big difference between soft and stiff at these higher levels of hormone. So we think it's important to have a stiff microenvironment in order to see the the interactions between the cells and the hormones. We also want to caution researchers that if you're doing sex as a biological variable in your studies and you're looking at cells from patients of different sexes, growing all of your cells in FBS might be a mistake because FBS is very high in progesterone, which had a big influence on our cells, especially the male cells, as you can see here. So I hope, based on all of that information, that I convinced you today that it's very important to study cell matrix, and sex differences when you're looking into modeling idiopathic pulmonary fibrosis. We're trying to move in this direction and to move into fully human systems using induced pluripotent stem cells. And we plan to continue making alveolar models and also work together with a collaborator in Iowa to make airway models so that we can investigate this link between genomic cell origin and the impact of circulating sex hormones in pulmonary fibrosis. Where are we going with this? Well, we have a couple patents that have been initiated or in progress. Every once in a while, I talk to a company that's really interested. So I wanted to show you, this is a little bit of our competitive matrix that we like to show to people when we're pitching our technology. So I say multicellular 3D models are an area that's really promising tool for drug discovery and preclinical testing and validation of those drugs. If we look at our models, like I mentioned, we have control in 3 dimensions. We can use all human cells. We can study cell-cell interactions and cell matrix interactions. Organoids are very similar, except they're mostly grown in matrigel, so it's much harder for you to study these cell matrix interactions. In rodent models, you can't have human cells, and it's very difficult to study individual cell-cell or cell matrix interactions. And of course, in a Petri dish, you're no longer in 3 dimensions. And again, you're usually culturing 1 cell at a time, so you're not studying these last 2 things. Organoids are a large and growing market. The global organoid market in 2019 was almost $700 million. It's projected to reach $3.4 billion by 2017. And only 4 companies produce in vitro lung models. There's a huge opportunity for growth because investment in personalized drugs is expected to grow 33% over the next 5 years. And we believe that our in vitro lung models can be used to start the process of understanding personalized drugs. So with that, I'd like to thank the folks in my lab who have worked on the research. I showed you research from Rachel Blomberg, Michaela Mueller, Alicia Tannenberger. We've also worked closely with David Riches, Amy Ryan, Daryl Weiss, Daryl Cotton and Daniel Weiss. I mixed those 2 together. And I'd like to acknowledge all of our funding sources. So thank you all for your time.
Thank you, Chelsea. Very interesting presentation. So let us see if we have some discussion from the audience. Any questions?
So there's one in the chat. It asks, what methods are being used to detect the pulmonary diseases of the lungs? Is computer vision, deep learning used for the prediction? If yes, how is the data set evaluated and gathered?
That's a great question. I don't know for sure. I know a lot of these lung diseases will be first identified just on an X-ray, and then I think further investigated with CT. I've been working with a team at National Jewish Health that includes Caroline Kuhl, who is a pathologist, and Kevin Brown, and they have sections of lung tissue that have these fibrotic foci in them. And we're currently working with Brad Smith in bioengineering to take pictures of these sections, stack them up, align them, and segment them to look at the fibrotic foci inside. But when we've talked a little bit about trying to use some machine learning techniques to do that, but I haven't gotten there. I don't think the lung field is quite there yet either.
Another question that came in is, you mentioned you have several patents in progress. Which ones are you most excited about?
Well, I guess 2. One from my days at the startup company is on a wound dressing that we invented that uses topography to guide cells to the right place to help heal wounds. And then the other ones are the ones that are from my lab right now, because those feel really special. So we have a patent on the 3D lung models and another one on the hybrid hydrogels that are both in progress.
Since I have a question, since you worked in private as well as in the university setting, how different is it to get your property through that innovation, CU innovations, and how different is it to try and get through that process of making it yours, right? Protecting it.
Yeah, that's a tricky question. There are advantages and disadvantages on both sides. I think one advantage to being in industry in a small startup company was people recognized our intellectual property was really important, and we tried to cover it very quickly. I've actually been working with the same patent attorney since graduate school. I brought him on here at CU as well. And so they've done an amazing job of kind of doing that really quickly. One disadvantage in industry is, of course, you assign your patent to the company and you don't make any money ever, personally. In academia, working with CU Innovations, I think because budgets are limited, that it's a little bit harder to convince them to file the initial patents, which was tough for me. I was used to that. But the advantage to filing through CU Innovations is that they do have sharing agreements in place for royalties with faculty. So if your patent is licensed and you start making money off of it, you personally do get to make a little bit of money, and your department and your college make a little bit of money. And so that's a really cool advantage to working with the CU Innovations team.
And they work with both CU Denver and CU Anschutz, correct? CU Innovations, it's a centralized unit.
Yes, that's right.
Noah asked, why specifically did you use the young bovine blood to recreate the model again? Was it mostly the molecular similarities or something else?
Oh, no, this is just a standard that's used in cell culture all the time. Anyone who grows fibroblasts will use the fetal bovine serum in the media. So we used it as kind of the control, which is, this is the gold standard. This is what people are doing now. And then how does it change when we actually present the real human serum composition that would come from a person?
Rachel said, hi, Chelsea, amazing work and presentation. Have you left, that's a word I can't say, alveolar?
Alveolar.
Thank you. Microsphere model to potentially include an air-liquid interface in the future or stretch? Very exciting work.
Thanks, Rachel. We think about this all the time. Yes, question mark. I do believe we can include an air-liquid interface in our alveolar models if we get a little bit fancier with the material. If we used potentially some of our photodegradable materials to make the microspheres, and then also included some way to access it. I don't know, you could just jam a needle through the hydrogel, right? We could very quickly remove all those microspheres and then add air to the inside of the hydrogel. And then in stretch, I think that's also important. It's something we think about that is sort of a side project for the future. We need to design a material that has more elastic properties so we can stretch it more before it breaks, before we start to stretch these models. But that's a great question, because the air liquid interface exists in the lung, right? We're breathing and there's air inside one side of the lung, and then the other side of the lung is exposed to the inside of your organ. And then of course our lungs stretch constantly. So that cyclic stretch is really important for maturation of the cells inside the lung.
Hi, Chelsea. I have a question. I was asking which instrument you were using to test the to do the tensor test, or specifically I'm wondering how you get the Young's modulus, because the hydrogel you are using is basically solid. It's not liquid, right? So I'm not sure how you use a rheometer to get the modulus.
Oh yeah, it is a solid, and we do use a rheometer with a parallel plate 8mm geometry. We can apply a shear force and then measure how the material is responding. We end up with a shear modulus G prime, and then we convert that to an elastic modulus. We've also verified that we can convert it to an elastic modulus using rubber elasticity theory by measuring the elastic modulus with an AFM as well.
Oh, okay.
Is that helpful?
Yeah. Because I was doing some tests on a hypoelastic material, and design the grip to do the tensile test is a challenge. So I just wonder, I guess that's a really good route to do the share modulus, to convert share modulus to Young's modulus, to elastic modulus. Yeah, that's really a good way. And also, I'm wondering, have you heard about a VXL printing, like a 3D printing, but the printing in like small VXL instead of a whole bunch of stuff? Is that helpful to build the long model?
Oh yeah, so great questions. I just wanted to comment on the conversion of shear modulus to elastic modulus. Make sure you understand the Poisson's ratio of your material, because that'll be the biggest factor when you do that conversion. And then asking about voxel printing. Yes, I think that's really, really quite exciting. We haven't gotten there yet in my lab, but I know at Boulder, Rob McLeod's doing a lot of that work. And I know that they've been printing materials like PEG-norbornene that we've used in our models. So it's definitely an area that I want to explore more. I haven't done yet.
Okay, I'm from Kai's lab in a mechanical engineering department, and we do 3D printing. So maybe we can calibrate one day.
Sure, that'd be great. Yeah, definitely send me an email.
Thank you.
So Chelsea, I have one question. So I understand that you've been working with medical doctors and patients as well, right? So you're an engineer, I'm an engineer. So I just wanted to ask if you could share your experience, working with the medical professionals as well as people, because we most of the time don't do that in our research, engineers in general.
Yeah, that's a great question. And, I think working in industry probably taught me this the best, but especially biomedical engineers, we tend to think, oh, I can make this great solution for this problem that I know is out there in medicine. But if we don't talk to the physicians, we don't know what they really need. We don't know if they would actually use our product if we made it. We don't know if the hospital would buy the product if we made it. So those interactions where you are able to meet and talk to physicians that are treating the patients that you are interested in treating is very, very important. And I think that could be expanded to all different types of engineering. If you're a civil engineer and you want to make a road a certain way, are people going to want to drive on it? Right? Or you're a mechanical engineer and you're making a car, or are people going to buy it? It's really important to understand who your stakeholders are and who your final users of your product would be, and to talk to them while you're designing things. I can say in interactions with the medical field can be challenging. Medical doctors are very busy. Some of them are not that interested in talking to you. And so you have to find, this is my advice in all collaborations, find the people you really enjoy working with, and be productive in that sense. You'll definitely find people that say no, but keep asking, you'll find people who say yes.
Thank you, appreciate it. Any other questions for Chelsea? Well, if not, I'm gonna thank you here again. Very interesting presentation, Chelsea. And thank you so much for taking time from your busy schedule to give this seminar to the students. I'm sure they appreciated that. And before we quit, I just very quickly wanted to, again, share the schedule with the students. You guys can see the Excel sheet there?
Yes.
So then our next seminar is on April 4th, and then we will have 2 more seminars after that before the semester ends. So I look forward to seeing you all on April 4th at 11 a.m. Of course, Tammy will send the invite, but I just wanted to make sure you guys know so you can plan accordingly. Okay? Yeah, so if nothing else, then we will finish right now. So thank you so much. And